Hi, thank you so much for the kind introduction and for honoring me with this award. It's really hard to overstate how important SMBE has been to me as a scientist, so it means a lot. So, since this award is named after Allan Wilson, I thought I'd start by motivating with what I do by what I've learned from his really foundational work on the molecular clock. So, one thing that really spoke to me in his sort of perspective piece on his clock work that he co-wrote in the 80s was that sort of until molecular clockwork began in the 60s, evolutionary biologists were sort of hamstrung in a way he compared to biochemists having to work without wall clocks and still test theories about enzyme kinetics, where maybe a wall clock it sounds kind of boring and pedestrian, but its importance is hard to overstate. And Wilson really demonstrated this utility of the molecular clock by using albumin to date ape divergence and mitochondrial DNA to date human diversification. But then the second point that he brought up was that we can't just sort of take for granted the utility of molecular clocks. We also need to think hard about why it is that molecular clocks work as well as they do. He seemed kind of amazed that this was the case, saying the existence of nearly universal molecular clocks provides a challenge to explain them as well as an opportunity to use them. And of course, you couldn't have this kind of molecular clock without a mutation rate that stays relatively constant over time. And what my lab is really interested in is digging into, to what extent do mutation rates evolve slowly, and why is that, and when does it break down? So something that's been really influential to my thinking about this is Mike Lynch's drift barrier hypothesis, which basically posits that mutation rate is like any other trait that evolves under stabilizing selection. And a way we can understand this is to think about organisms devoting a certain number of resources to avoiding DNA damage. And the more resources you devote, the more your mutation rate goes down. But at some point, the marginal benefit of avoiding more mutations won't be worth the marginal cost of spending more resources avoiding them. And so then there will be this biophysical optimum where the marginal cost equals the marginal benefit. But now if we stop thinking like an economist and start thinking more like a biologist, we can think, okay, say a population achieves this optimal mutation rate and then has like a mutation in a DNA repair gene. Well, if the mutator is strong enough and creates strongly deleterious mutations, it'll be selected against, and the mutation rate will sort of go back down again. But perhaps weaker mutators might not have this happen to them and might segregate nearly neutrally. And this is especially likely to happen in sexual organisms because so many deleterious mutations could just recombine away quickly from any mutator that created them. And the upshot is that populations, Lynch predicts, will exist at mutation rates somewhat above the biophysical optimum, and how much above depends on the efficiency of selection as modulated by the effective population size. And so one prediction of this is that humans and other apes with our relatively small effective population sizes compared to organisms like Drosophila or bacteria will have relatively suboptimal mutation rates. And also relatively less constrained mutation rates. And that makes it especially amazing that it worked for Allan Wilson to assume that these molecular clocks would hold and find evidence that they held reasonably well, and be able to use them to date interesting events in our evolutionary history. So how could we test the predictions of this drift barrier hypothesis and maybe estimate the strength of some of the parameters involved? One way would be to directly measure de novo mutation rates in a bunch of organisms from similar and different populations and just directly look for differences. And this has been done to some extent, but it's still really expensive. And so what my lab has thought more about is sort of a more counterintuitive strategy that involves testing for differences between the spectra of mutations that are segregating in different populations. And by a mutation spectrum, I mean a histogram that sort of describes how mutations are partitioned into some different categories, usually what is the triplet sequence context that they fall in, and the ancestral and derived alleles. And the reason why this might be informative about evolution of mutation rates is that if mutation rates evolve as a result of mutator alleles, we know from work on mutator alleles in things like cancers and microorganisms that they tend to disrupt specific pathways and then cause mutations of particular types. They don't just sort of cause more mutations of all the types in the proportion that they already occurred. And so then the types of mutations that a particular mutator is most likely to create form a distribution known as a mutational signature. And so one of the most sort of influential contexts in which mutational signatures are talked about is in the case of cancers, as pioneered by Alexandrov et al. In 2013, and shown by this example of a signature of a mismatch repair deficiency in this 96-dimensional triplet space. So when I sort of had this crazy idea to look for differences in the triplet mutation spectra of different human populations, I found sort of a more dramatic result than I ever expected to find, which was this global imbalance in the proportion of mutations from TCC to TTC. So this proportion in South Asia and Africa it makes up about 1.5% of all mutations, but then in Southern Europe, it is close to 2% of all mutations. And that 0.5% difference is hugely significant when you think about how many millions of private variants have now been ascertained in all of these different human groups. And I think it's also a difficult pattern to explain using only sort of the normal population genetic processes of things like selection and demography. So this, in a nutshell, was what convinced me that, mutation rates do evolve in subtle ways over timescales of just a few thousand years in the cases of human populations. And it turns out that this isn't just a one-off difference. It's the most sort of dramatic one still that I've found. But if you look at humans and great apes, for example, then it turns out you can tell what population or species an individual is from just by looking at their mutation spectrum. And these qualitative results sort of tell us that there seems to be this mutation spectrum drift that's always going on as a result of mutator alleles emerging nearly neutrally and just causing population mutation spectra to do some kind of a random walk. But like a method developed by my grad student Will DeWitt has really started to put this on a more mechanistic empirical footing. So Will's paper MuSHI that he collaborated on with Cam Harris and Aaron Ragsdale was recently published and is available for anyone to try out. It's very versatile. If you can compute a site frequency spectrum from your data, then you can try MuSHI and probably discover something new and interesting. So in the human 1,000 genome population, what it gives you is these temporally calibrated trajectories where all human populations sort of start out in the ancient past with a similar mutation spectrum, and then they diverge from one another over time. And the way that MuSHI does this, in a nutshell, is to start with the site frequency spectrum with each category, singletons, doubletons, and so on, partitioned by triplet context. Is called the KSFS after a k-mer because it can also be adapted to k not equal to 3. And then you can visualize this as a matrix of site frequency spectra of a bunch of different k-mer types. And then the first thing that MuSHI does is to infer a global demographic history for each population that you're looking at. And these are like pretty nice PSMC style histories that actually come from site frequency spectrum data, which even if you're just interested in demographic inference, you might want to check out. But then the downstream thing that MuSHI does is to use this demographic history to then calibrate the time scale of change that is most likely to have led to differences in the site frequency spectra of the different mutation types. And it sort of uses some regularization strategies to find a relatively low rank small set of mutational signatures that sort of change over time and can explain these differences. So what MuSHI gives us is a really nice way to sort of describe like how fast populations are evolving in their mutational spectra over time, and sort of how many independent mutational signatures you have to think about. But it doesn't get to the causality of these things. And so I'm also going to present some parallel work we've been doing that's more motivated by the question of, like, are these changes driven by genetics or the environment? And if genetics, what sorts of genes might be responsible? And what might they mean pleiotropically? Like, is there anything that's happening to the health of these populations as these mutational signatures go up and down in intensity? So one thing that sort of solidified my hunch that genetics might be responsible for a lot of this was an exciting paper by Beth Dumont from the Jackson Lab. So what Beth did was she looked at rare variants in the Sanger lab mouse strains. And these variants likely accumulated while these mice were living in captivity. And so they aren't likely to have been infected by really many differences in things like diet or stress or environmental exposures, and yet you still see quite a bit of variation of mutation spectra among these different mice that loosely segregates by how closely related the strains are to one another. And so we think these differences in strain-specific spectra are likely not driven by anything environmental. And they're also not likely driven by any kinds of demographic effects like generation time differences, because these mice are told sort of when and where to breed. And so you don't have the uncertainty that you have in natural populations like humans of not really knowing at what ages people reproduced in different populations, and us knowing that indeed paternal and maternal age can have effects on the mutation spectrum. And a thing that kind of pleases me about this study is that all these mutations that Beth got these insights from are sort of an annoyance in the original context of breeding all these mice. Like, you inbreed mice for various purposes, but then that causes mutation load, and then that's regarded as this fly in the ointment, which luckily we now have a use for. So after seeing these mutation spectrum differences among lab mouse strains, of course, one of our first thoughts was, could you do some kind of QTL mapping, crossing together mice with different mutation spectra and then looking for genomic regions where having a particular parent is associated with some mutational phenotype? And luckily for my dry lab, we didn't have to do this ourselves, but we're able to collaborate with people who've worked for a long time on a data set called the B by D, or BXD, this really exceptionally old, large recombinant inbred mouse collection that was actually founded over a series of epochs, starting with Ben Taylor at the Jackson Labs in the 1970s. And there's a total of 151 of these lines, which were all generated as either F2s or advanced intercross lines from a black 6 parent, a C57BL6J parent, and then a DBA2J parent. And these are lab strains that are about as diverged as European and American humans. And then the oldest of these lines have been propagated by brother-sister mating for up to 188 generations, sort of accumulating mutations every step along the way. And the reason that people have been accumulating these this whole time and creating sort of this natural, well, not natural, but say inadvertent mammalian mutation accumulation line set is that these BXDs were really useful for doing QTL mapping of other tricky traits, sort of like drug and alcohol dependence. And so we were really hopeful that they could help us with the tricky mutation rate trait too. And here's just flies in the ointment, accumulating at every stage of the breeding. So luckily for us, the parental strains that Ben Taylor chose are ones that Beth determined to have different mutation spectra. And together with a small consortium, we sequenced all of these strains to 30x coverage. And then my postdoc, Tom Sassani, who did the bulk of the computational work on this project, went to work calling all the homozygous singletons, so variants that are fixed in a particular line and absent from all the other lines. And a preprint on our work is available for you to take a look at. So our hypothesis was that all these homozygous singletons would be mutations that occurred sometime during the inbreeding and fixed in the line. And sort of in support of this, we see that the cohort 1 that's been inbred for the longest time tends to have the most mutations. And then these all just have so many more mutations than you ever get in a standard trio, because of course they've accumulated over lots of generations. And another nice thing is, since these are homozygous, you don't have to deal with as many of the bioinformatic headaches that come up determining whether true de novo mutations are real or not. So of course, the first thing we did with these data were to divide the number of mutations by the generation of inbreeding as an estimate of mutation rate and do a QTL analysis on the total rate. And for that, we didn't get any genome-wide significant hits. But then when we measured, when we did a QTL analysis on the rate of each mutation type separately, we found a highly significant peak on chromosome 4 associated with the C A mutation rate. And this peak becomes even more pronounced when you instead look at the C A fraction. And it's actually the DBA2J ancestry at this locus that's associated with higher C A fraction genome-wide. So one thing we did was, of course, just look at the C A fractions of all the D haplotype mice and the B haplotype mice, and the D haplotype C A fraction is around 22% on average, where the B haplotype one is less than 15%. And then when we did a principal component analysis of these different of the mutation spectra of these lines, we saw that whether or not you have this QTL haplotype explained the first PC pretty exactly. And when we looked at the triplet context, the triplet context in which the D haplotype was most enriched for mutations, we found this signature dominated by CA to AA and CT to AT mutations. So the next thing we really wanted to do was look at this 5 megabase QTL interval and see whether we could find map what gene in this interval might be responsible for the phenotype. So it contained about 100 protein coding genes total, but only 22 of them contain any variants between the B and D haplotypes annotated as moderate or high impact. And then only one of those genes was involved in DNA damage response or repair. And that happened to be this gene called MutYH, which we were very excited to see is involved with repairing 8-oxoguanine lesions and preventing C to A mutations. So it turns out that some research that researchers led by Yusaku Nakabepu in Japan had generated MutYH knockout mice and observed that they had an extremely elevated mutation rate dominated by C to A mutations. And we found that the signature enriched in these knockout mice was pretty much a dead ringer for the signature we found as enriched in the D haplotype mice relative to the B haplotype ones. We also found that there's a human signature associated with a MutYH variant. Basically, there are some rare variants in MutYH which, when homozygous, make people susceptible to this rare early onset colorectal cancer syndrome called MutYH-dependent polyposis. And colorectal tumors isolated from these patients have a high load of this C A dominated signature called SBS18, which also is highly correlated with the signature that we found in the mice. So although we haven't sort of done a CRISPR experiment to tell that MutYH is the gene responsible, we think this is pretty strong circumstantial evidence. And when you go and look at MutYH and count the differences between the B and D haplotypes, it turns out there are 5 missense differences, which where we think any of these could be responsible for the QTL. We haven't fine mapped it to one of them. But one thing that we were very excited to see was that all 5 of these variants are segregating in wild mice, both in the Castaneus subspecies and the Domesticus subspecies, from which most lab mouse variation was originally derived. And then when you look at Mus spreadus, which is a 2 million year diverged out group, you kind of see that it looks like 4 out of the 5 of these sites, the DBA2J allele is the ancestral allele, which was not what we expected, since remember the D allele is the one with the higher mutation rate. But it seems like maybe these mice were happy hanging out at a suboptimal mutation rate for a while, and then had this mutation that was then positively selected, which then was able to improve the mutation rate by like a small but noticeable amount. So perhaps that supports the drift barrier idea of populations hanging out at these suboptimal mutation rates. So finally, we looked back at the Sanger mouse genomes that Beth Dumont had studied, and we found that there's some breakup of the LD of this haplotype, where a subset of the Sanger mouse share 3 out of 5 alleles with DBA2J, and the rest with BLAC6. And then when we looked at that group, we found they had a significant C to A difference from the alleles that shared all 5 with DBA2J, but not a significant difference from the group that looked like BLAC6. So that makes us think that narrow it down to potentially just 2 of the 5 alleles as the most likely one to be responsible. And then the triplet context of the enrichments in these Sanger mouse is noisy, but does look vaguely like what we see in the B versus D haplotypes. And then finally, a multiple sequence alignment does support the idea that it's a derived anti-mutator instead of a derived mutator. And I'll just close by saying, if any of you have some recombinant inbred lines lying around from any model organism, the older and sicker, the better. You should sequence them and try to find a mutator of your own, or collaborate with us to sequence them. We'd love to do more of these studies. So in my last few minutes, I'm going to switch gears to a different model organism, yeast, that has been the focus of work by my fantastic postdoc, Pengyao Zhang, who, by the way, is going to be on the job market next year. So what Pengyao has been studying are these 1,011 yeast genomes, which were isolated from many different natural and domesticated environments. And as we usually do when we look for mutation spectrum variation, we found it in this data set. And what Pengyao did was she devised this really powerful experimental setup to test whether any of this variation was driven by extant mutator alleles whose activity we could detect in the lab. So she worked with Maitreya Dunham and her student Anya Oladart to design this sort of modification of the classical Luria-Delbruck fluctuation assay that would help us measure mutation spectra of a lot of these strains. So it involves just growing up these yeast and then using cannabinine to select for ones with mutations in CAN1, but then growing up the mutants and sequencing their CAN1 to figure out what type of mutation it was. And this is just a lot higher throughput than something like mutation accumulation lines. And what we found was that 2 strains that both come from the Mosaic Beer clade had mutation spectra with a pronounced C to A skew, and also the highest mutation rate of any of the strains we found, about 10x elevated over standard lab yeast. And then when we looked at the rare variants among all of the yeast and did a PCA on them, we found that these same strains were enriched for C A. So concordant evidence of the same mutator phenotype in the lab and in the variation. And then when we looked at the sister lineages to these in a couple of different yeast phylogenies, we found an elevation of the C A fraction among rare variants, suggesting that these other guys share the same mutator. Finally, we found that AAR and AEQ, these two yeast strains, had a shared non-synonymous variant in a DNA repair gene called OGG1, and to test whether this variant might cause their phenotype, we cloned it into a plasmid and used it to transform an OGG1 knockout strain that was otherwise identical to standard lab yeast. And we were really excited to find that this only was able to partly complement the mutator phenotype of the OGG1 knockout. So basically this shows if you knock out this DNA repair gene, you get a high mutation rate compared to, and then if you sort of transform that yeast with a plasmid containing standard lab yeast, OGG1, you get a low mutation rate again. But then if you complement it instead with the Mosaic Beer OGG1 variant, you get an intermediate mutation rate. So this allele looks to be subfunctional compared to the one that we usually see. And then the sort of cherry on top is that invertebrates OGG1 and MuTyH are part of the very same 8-oxo-G repair pathway. So, I mean, these somehow, we're very interested to see whether this sort of pathway shows up again and again as sort of breeding ground for mutator alleles. So just an emerging takeaway, I think, that we're really interested in exploring further is this idea that there's a lot of pleiotropy, seemingly, between cancer risk and variation in germline mutation spectra. So we've learned, I think, several things supporting this, and also have a lot of open questions to explore. So one thing we've learned are just that there exist these mutator alleles that are relatively common in MuTyH and OGG1 in mice and yeast, respectively, and that both genes cause human cancers. And then a final thing, not from my lab, but from really exciting recent work from Matt Hurls's and Peter Campbell's labs, is that there are rare human mutator phenotypes caused by cancer-associated genes, as shown in this plot of mutation rates versus paternal age, which has some extreme outliers. But still open questions. I really want to return, of course, to this TCC pulse that started this line of work. We still don't know what could have caused it, genetics or environment. And also with the other human mutation spectrum gradients, how many of them are genetically determined? And then what does that mean for human disease phenotypes? Does this tie into cancer risk in some way? So I'd just like to acknowledge everyone in my lab and all our collaborators who made this work possible. Say we're looking for postdocs. If you want to join us, please get in touch. And thank you again to SMBE.